Yo,
Hello.
how are you? Um, say something? Oh, can't hear you.
Tutor.
Yep.
Okay, yeah, I feel like I always try to join Zoom with my headphones, but it never works for some reason. So
Oh, that's weird.
I know, I don't know what it is every time. So I'm just like, fine, like, I guess it'll be out loud.
Got it, got it. Yeah, all right. What's new? How are you?
I'm good. I can't complain. I took my for the AAMC FL1 last week, and I did like a little bit better than I did on the FL5, so that's a plus. But I'm still like not necessarily where I want to be. But I'm also not too discouraged because I improved in cars. I got like a
Uh,
50.
sorry, one second. I just want to.
Okay.
Okay. What were you saying?
I said but I'm not like a little too discouraged because I got to improve in cars. Like I got a 50% this time instead of where I'm normally at. So we're like steadily improving in terms of cars. So that's
Totally.
given me a little bit of faith. And I reviewed my chem phys section and biochem section, which I also felt like I was doing. Like I got more questions right than I normally do in biochem. So I was like, I feel like I'm heading in the right direction. It's just like little things that I'm missing. Like when I was doing the review, like certain answers to the questions were in the passage, and I was just like God, like it's kind of frustrating. Like when you
Uh-huh.
miss it, but it's like okay, going back the second time, and you're like, oh, okay. So there was like a couple questions like that for me with biochem and chem phys. I think I just need to work on a couple more things. Like I don't necessarily know about I watched a video on it, the converging and
That,
concave lenses that
yeah.
um and then there's like the what's it called? Radioactive decay. And yeah, there's just a couple of things for chem phys that I need to touch on. But I don't know. I feel like as I keep doing practice, I like now I'm trying to do at least like 60 practice questions a day. I did 40 chem phys questions the other day. I didn't get to do too many biochem questions yet, but I'm gonna do that after we get off of the this Zoom. So yeah, that's what I'm just trying to just keep drilling it until I understand.
Got it. So, what would you like to go over today? Optics and radioactive decay or some other stuff?
So I wanted to go over passage 8 of the Campus section for AAMC Fl1 just because that one I had the most wrong, and then when I was reviewing it was kind of confused, and then there was like that I guess optics question.
Sure.
Yeah. So let me pull that up and then share my screen.
Hmm.
How have you been?
I've been good.
Okay.
Um, yeah, thank you.
Yeah. Okay. Like I guess I'll just do review all with Jack Weston, hide the solutions. Okay. Okay, this one I shouldn't have got wrong. I did this question over again and I understood it the second time.
Uh-huh. Okay. So this is the passage where you got a lot of questions wrong.
Yeah, like I
Okay,
didn't.
so let's have you read the passage and yeah, we can talk about um we'll probably be writing like proportionalities and stuff. So but yeah, go for it.
Glycogen synthase catalyzes the lengthening of glycogen polymers through the addition of glucose. The reaction consists of transferring glucosyl moiety from uridine diphosphate glucose to the non-reducing end of the glycogen molecule along the release of UDP. UDP glucose is formed from the enzymatic.
Sorry, so what are they telling us so far?
Um, so glycogen synthase, it um, works to add. It works to lengthen the glycogen polymers, like by adding glucose.
What is that?
And there's glycogen. It's stored in the liver, and it's I guess like it's just like the stored form of glucose in the liver.
So um okay, yeah. So glucose would look like something like this. I mean, like with the OH groups and stuff.
Hmm.
Um, so this is the so something like that. First carbon, second, third, fourth, fifth, and sixth, right? So you could have bonds to another glucose that could be between the first uh the first carbon position of one glucose monomer and attached to the fourth carbon of the other one, and that's your alpha 1,4 linkages, which are linear, they're in a straight line. But you could also have,
The branching.
yep, where it's a 1,6 linkage, right?
Hmm.
And they're going to be alpha because we as humans lack the enzyme to break the beta linkages, but that's something like, cows and like animals, horses and stuff can do. Um, so okay, so glycogen is found in animals and it's the polymer or polysaccharide form of glucose. And yes, you have those 1,4 linkages and you have the 1,6 branching linkages. And when they say this non-reducing end, what do they mean by that?
It doesn't have a it doesn't have a negative charge, is what I was assuming.
So this is something that they like to ask you about in sugar stuff. So basically, and then also for every biomolecule, you want to be able to describe the bond in terms of like organic like chemistry functional groups. So for instance, um, this here is so I can consider this an OR group, R group, and then hydrogen group. So that's a glycosidic bond. But if
Mhm.
you have a like monomer of the glucose, this then would be this functional group. Now, do the names of any of these ring a bell?
Um, there's a hydroxyl group
Okay.
and I think that's a ester.
So, so an ester will, since it's a carboxylic acid derivative,
Oh.
would have to have a carbonyl group.
Okay, okay, okay.
But this is going to be called a hemiacetal,
Oh, okay.
right? And if this was an R, another R group, that would just be a hemiketal. And it's kind of just like how, this is an aldehyde, this over here.
Yeah.
And if this H was another R group, it would be a ketone, right?
Mm-hmm.
So same kind of thing here with acetal being like the aldehyde form, ketal like the ketone form. Uh Now you should also know like the generalized biochem redox, like what that looks like. I have like in the PDF stuff folder like a document that's just called or a PDF that's just called biochem redox, but you'll see that because when we talk about redox in Gen Chem, it's like change in oxidation states, but in biochem, like that's not really what you see. Instead, you see either loss of hydrogens, addition of oxygens, or addition of bonds to oxygen.
Okay.
So that means that an alcohol group, which would be something with a hydroxyl, can be oxidized further into an aldehyde or a ketone. Um, and so if something is a reducing sugar, it reduces other things, right? Which means it itself gets oxidized, right?
Yeah.
So that's something where, so like a hemiacetal or a hemiketal, since it has the hydroxyl group, could be considered a reducing sugar. Um, but when it forms a glycosidic bond and it turns into a like instead of a hemi, it'll be like an acetal or a ketal. You lose the hydroxyl group, and therefore you lose the ability of it to get oxidized, which means that it loses the ability to be a reducing sugar. And so basically, if you have a monosaccharide, you will have that reducing sugar. If you have a polysaccharide, you'll get rid of it, but at the very end of the polysaccharide, you will still have the hemiacetal hydroxyl. So even a polysaccharide can test positive for reducing sugars because it still has just one reducing sugar. Um, and also they like to ask about Holland's test and Benedict's test, which are tests for reducing sugars. So Toland's test involves it's called a silver mirror. It's like solid silver to form. And Benedict's test has a copper like brick red precipitate form, and both of those are positive tests for reducing sugars. So here, where it tells us that this reaction involves transferring a glucosyl like group, which is just like a glucose group,
Hmm.
from UDP glucose, which they give us a picture of, and they say that it's added to the non-reducing end of the glycogen molecule. So if this is the glycogen molecule, right, this would be the reducing end. So it would add somewhere here instead, then, right, for the
Yeah.
non-reducing end.
Yeah.
So so those are just some buzzwords that you'll see like as you start to read this passage, and like it doesn't mean to like stop and go over it like the way that I just did, but these are just things that you want to have like pop up in your head uh because they're things that they'll pretty much likely be asking you.
Gotcha.
Mhm.
It was discovered that approximately one out of every 600 to 1500 glucose molecules in muscle glycogen contains phosphate groups covalently linked to the C2 and C3 positions as monoesters. Mm. It is hypothesized that glycogen synthase is responsible for the phosphorylation. Um, I think this is just giving us information that scientists have found. And I guess the theory is that glycogen synthase is responsible for it.
Okay. So then we see our molecule, and
Yeah.
whenever you see the structure of a molecule, you want to identify functional groups. So what do you see here?
I see phosphate groups, glucose, hydroxyl groups. I also see, is this a ribose sugar?
Yep.
Um,
That'll just be like a nucleic, like a base, right?
oh yeah, that's the uridine.
Yep. And it's essentially just like a type of like thing with like uracil, right?
Yeah.
And makes sense because since we have two hydroxyl groups on the ribose, that wouldn't be deoxy, right?
Mm-hmm.
Just a regular. So okay, good, good, good. Yep, you can continue.
Lafora disease is a fatal progressive epilepsy that is characterized by the formation of deposits of sparsely branched, insoluble glycogen-like polymers called Lafora bodies. So, this is talking about a disease that is caused by the glycogen-like polymers, and they're called Lafora bodies.
Yep. And so so the reason that glycogen is branched, by the way, is because it increases the surface area and thus makes it more soluble. So, so now if they're telling us that this disease involves less branches, oh, I think say something.
No, no, no. I
Yeah.
was just whispering to myself because I missed
Yeah.
honestly. Sparsely, yeah.
Oh yeah, yeah. So um so it should make sense then that if there's less branches, it'll be less soluble, right?
Mm-hmm.
So good. So it seems like so like things that you can think about like so far would be like, okay, they'll probably ask me about those 1,6 linkages,
Yeah.
right? So like we want to be able to read actively like this so that and also be like priming our brains to like think of these things like these associations as we see the words. And yeah, like when you do it enough, you'll be able to like predict most of these questions. Yeah.
Gotcha. LBs contain a much larger amount of covalently linked phosphate compared to normal glycogen. The abundance of phosphate has been linked to a mutation in the gene for Laforin, a phosphatase specific to carbohydrates.
Okay. What are they telling us here?
It's saying that the disease, well, the Lafora bodies that like come with the disease have a lot of covalently linked phosphate compared to like the normal glycogen,
Okay.
and it causes a mutation. Well, I guess not linked. It's not caused. It's linked to a mutation in the gene for Laforin,
Yeah,
which is
probably the other way like around, right? Like the
yeah,
mutation can cause because all right, yeah, tell me about the mutation.
uh, it's in the gene Laforin, which is a phosphatase specific to carbohydrates.
So what is the phosphatase?
It gets rid of phosphate groups.
So this mutation, do you think it makes more of the phosphatase or less?
Makes makes more.
Uh-huh. So we could say that Lafora disease, or I'll just say Lafora bodies, are inversely proportional to the alpha 1 6 glycosidic bonds, meaning less branching,
Hmm.
meaning less solubility,
Hmm.
also meaning more phosphate. I usually just draw like P with a circle around it. Um, and I'll say also means less laforin or phosphatase, right?
Yeah.
So so basically like we are able to link all of this stuff together from what they tell us. And when we do the proportionality, it makes it really simple to see the relationships and helps us not, get lost in all of like, the words and stuff like covalently linked,
Mhm.
like like phosphate is always going to be like covalently linked and stuff. So we see that okay, this disease it means that we have less branching, which means we have less solubility, and they noticed that this occurred when there was more phosphate, and it's linked to this mutation for a phosphatase, and we can, even though it doesn't tell us, we can infer that if the phosphatase removes phosphates and phosphates cause this Lafora body, then the mutation probably causes like the phosphatase to get messed up.
Yeah. And yeah, the increase or
Hmm.
more. In order to test if glycogen synthase is responsible for glycogen phosphorylation, both UDP glucose and beta 32 phosphorus, UDP glucose, was incubated with muscle extracts from the wild type mice and mice lacking muscle glucogen, no, my bad, glycogen synthase.
Okay, so when they say that beta, what does
Hmm.
that
Um, I was thinking it was like the radioactive decay.
mean? Yeah, so that's what they want you to think.
It's not it.
So, now, did I go over with you like the reaction with like a kinase is like STY and phosphate, like how it phosphorylates, dephosphorylates stuff. So,
Mm-mm.
so like a kinase uses STY, like serine, threonine, or tyrosine because they have hydroxyl groups. And
Hmm.
those, whenever you see that, think, nucleophile, and a phosphate is going to be an electrophile. And as you move outward from like ATP, for instance, you're going to count in Greek outward. So, like, for instance, in this structure here, this phosphate will be called the alpha phosphate. This phosphate is the beta phosphate, right? And, if there was a third one, like there would be an ATP, that would be the gamma phosphate, the outermost one.
Oh, okay, yeah. Makes
Mhm.
sense. Okay, so that's what they're talking about. That makes okay. Wow.
Mhm.
Okay. The reaction products were separated by SDS-PAGE and visualized by autoradiography that detects the products of beta decay.
Ah, look, yeah. So now, okay, I can probably see what uh, could have gone wrong here. So we have both the radioactive beta decay, like you mentioned, and the beta phosphate. So yeah, that can be confusing. Yeah. So okay, tell me about beta decay.
That one is, it's when you lose a neutron, or you lose something. I'm not quite sure, or a positron, one of the two.
Can you hear me?
Yeah, I can hear you.
Yeah. There's some people playing really loud music outside because I guess work hours are done, so I need to wear this. Sorry. Okay, what were you saying?
I said, I feel like beta decay is the loss of a positron or something.
So, so we could have a beta plus be the positron.
Oh,
This
okay,
one
okay,
is the beta minus, and that's really just essentially an electron.
okay.
Yeah. Um, okay, so it seems like so yeah, so they're starting this experiment and they have labeled like okay, so we have the UDP glucose structure here, and so they labeled like one of the carbons of the glucose part, and then they labeled wait, both this glucose and this. Okay, yeah. So they labeled a carbon on the glucose. Wait, hold on. I keep UDP. Yeah, okay, okay. Sorry. So yes, this is the label of a carbon on the glucose, and then
Mm.
this is a label on the phosphate, on the phosphorus atom, right?
Yeah.
Okay. And they incubated them with muscle extracts from wild type mice and mice that have the knockout of glycogen synthase. Um, and then they did SDS-PAGE and some, visualization using products of beta decay. And yeah, continue.
And the data were normalized to account for the different decay rates. The samples were also treated with glucose sidase, an enzyme that breaks down glycogen, and assayed in the same manner.
Um, okay. So, yeah, all right. So now take a look at this figure and tell me what you see.
Um, so this is the, you said it was this one, right? This glucose that was labeled? I guess that doesn't necessarily matter. So this is like the decay results via the SDS page for the carbon 14.
Mhm.
And
Yep. And then the plus and minus represent whether um, glucosidase is there or not.
oh yeah, yeah, my bad. Um,
So, yep.
so the wild type mice still has the I guess there's no beta decay happening within the wild type mice without the glucosidase. And the muscles without glycogen synthase, I feel like they don't have, well, they have a little bit of decay, I feel.
Okay.
I think that's what it's saying. I feel like I honestly didn't understand this figure, which is why I was like,
Sure,
but
sure.
for
Yeah. Oh, yeah. Go on.
was gonna say for this one, I feel like it's a similar result, but the band is a little thinner for the phosphate group.
Okay. Yeah. Okay. Okay. So the glucosidase would break down the glycogen, and the glycogen synthase is what would build the glycogen. And for the MGSKO, they lack the thing that builds the glycogen, right?
Yeah.
And so yeah, for like figures and stuff, like I always favor like a relatively slow systematic approach.
Okay.
And so I would just look at it like, okay, wild type without glucosidase tests positive for this carbon, which is the label that they put on glucose.
Hmm.
So it would make sense because um, we don't have the thing that breaks down the glycogen, and we do have the thing that makes the glycogen. So yeah, we're going to see some glucose come up. And then when we have the plus sign, we have glucosidase, and that should break down the glycogen. And it doesn't seem like we have that what is it? The radiolabeled glucose. And then for the knockout, we have, I guess, a small amount of the um carbon labeled when we do not have the glycogen synthase. Then when we have the glycogen, oh sorry, when we don't have the glycogen synthase or the glucosidase, and then if we don't have the glycogen synthase or the glucosidase, then we shouldn't see anything, and we don't. Then we see similar results on the right, but they're a little thinner. And it seems like, let's see. Okay. Yeah, I was going to say that if we break this out, you would still have these. And I was trying to think if that would play a role or not, but yeah, I think that's, I guess, what we can take from this. And yeah, on the test, when it comes to these figures, I would spend like 10, 15 seconds to kind of like try to understand what it's trying to show you. Um, but if anything takes longer than that, just move on and go back to it if they ask you like a question about it, because either they'll ask you something about it that's easy to answer, or they won't ask you anything about it. So, yep.
Gotcha.
But yeah, writing out this proportionality, I think, is going to really help us, as well as like what we were saying before about the buzzwords helping you learn to anticipate questions. So, so okay. Any questions so far?
Mm, no.
Okay. All right, let's try this question out. And it's kind of like we kind of talked about this, but yeah.
So I feel like it's the alpha 1,4 glycosidic bond.
Mm-hmm. So tell me how you know that.
Um, because the glycogen synthase branches together the glucose molecules um by adding it to the non-reducing end of the glycogen molecule. And
Mm-hmm.
yeah, and then because then it's like the alpha, I think you were saying is has to do with the phosphate.
Mm-hmm.
So I feel like it attaches to the alpha phosphate and not the beta.
Okay. So I think for this question, what they're trying to do here is to have you see, and you remember how I told you like how people do cars and stuff? Like
Mhm.
people a lot of times like would do that for like passages like this too. Like it's like, okay, they said beta, so maybe the answer is going to be C or D. Did you choose something like C or D?
No, I think I put B. I couldn't remember if it was 1.6 or 1.4 during the exam.
I see, I see. So you knew it wasn't the beta.
Yeah.
Okay, that works.
Yeah. But yeah, it's A. Yeah. We'll figure it out. Or let me see what I picked. Yeah, yeah, I picked
Okay, you just said the 161. Got it.
Goofy. Um, oh, also I'm just like sometimes I get confused because like when I was first going over this exam, like it was showing me the exam that I took like 5 years ago, like when I first like was gonna take the MCAT. Um, so I would remember thinking like, oh, I think I put because I remember I was like, oh yeah, it's definitely ribose, and then I went to check it and it said that I had something different. And then when I came out of the review, it was like re me reviewing like not the test that I took last week, but the test I took 5 years ago. So I'm not quite
Oh,
sure
yeah,
why it keeps glitching like that. But I mean, I feel like this I knew it was ribose.
got it.
Let me see here.
Yeah, because so yeah, you don't really need to know like what xylose or arabinose look like, but
Hmm.
fructose you should know. And that would be
6.
uh was that
Oh, well, I feel like fructose has 6 carbons as well, right?
yeah, yeah. Um, but since it well, yeah, so since fructose has it's a ketohexose rather than an aldo, it would make a five-membered ring like that.
Oh,
As opposed like because okay, so like glucose has it's an aldohexose because the first carbon is an aldehyde.
mm-hmm.
And so when this closes the ring here, this is the fifth carbon's hydroxyl with making a bond with the first carbon's carbonyl. And so this would give you a six-membered ring like that, right?
Mm-hmm.
Now, if this was fructose, it would be it'll still be a hexose, six carbons, but it'll be a ketohexose, which means, and if we have a ketone, that means that the other end here must be the carbon. So for the ketohexose, this same hydroxyl, the fifth one, is going to attack the carbonyl like usual, but the carbonyl is now carbonyl carbon that is now at the second carbon position rather than the first. So this would create a five-membered ring.
Copy.
Yeah. So good.
Um, yeah, I feel like this is the question that I was confused on. Um, addition to of Laforan to the reaction mixtures used to generate the data in figure 2 will result in um, so it's a phosphatase
Mhm.
and well, this one, the knockout mice don't have any glycogen synthase in it, so there wouldn't be any glycogen phosphorylation in either of these. Um, just mainly looking at the wild type band. So I guess it would decrease. the intensity of the band.
Now, which band would that be?
Um, See, I'm not quite sure which band it is yet, but I was
So,
just thinking
so what does Laforan do?
it's a phosphatase specific to carbohydrates.
So, so what does it do?
It breaks down, it gets rid of phosphate groups.
All right. So we should see less phosphates, right?
Yeah.
Okay. So we should be able to get rid of A and B, right? Because only C and D talk about the phosphorus, the radioactive phosphorus.
Oh yeah, you're right. That's a good call, honestly.
Hmm.
Hmm. Okay, I see.
And yeah. So now now what can we choose?
Yeah, I would pick C that it would decrease the
Yep.
intensity.
Yep. And we see that connection here, right? It's an inverse proportion between phosphatase and phosphate,
Hmm.
meaning if you have phosphatase, you have less phosphate and vice versa. So yeah, good, good.
Okay. Oh, that's so, I feel like I just did not notice that. I mean, it's okay. That's why we're practicing.
Hmm.
Um, the radioactive decay described in the passage results in the formation of which of the two atoms? Yeah, I didn't know for this one. I just picked something random because I was kind of confused.
Hmm. Got it. So okay, let's talk a little bit about decay products. So your okay, so all of this really just comes from let's see how I should put this. So like let's say this is the number of protons, this is the number of neutrons, and they like the universe wants them to be equal.
Hmm.
Um, but let's say that you had something here which has more protons than neutrons. We call that neutron poor. And so what that means is a proton could decay into so um so a mass of a proton is equal to a mass of a neutron, right?
Yeah.
And we could say that as one,
Hmm.
like and the mass of an electron we can say is zero. And then we can say a proton has a charge of +1, an electron -1, and a neutron no charge, right?
Hmm.
Now, if you take so now a positron is essentially a positively charged electron. So it's going to have a mass of 0 and a charge of +1.
Gotcha.
So you could think of a proton as being a composition of a neutron for the mass and a positron for the charge, right?
Yes.
So a proton could break apart into a neutron and a positron. So if it was like positron decay, that would be for something that has an excess of protons or, you know, a deficit of neutrons. So something here will undergo the decay to go to the left and be on that line, right? And then the opposite is true if we had something here. If we had something here, we have more neutrons than protons. And so it's going to be neutron rich. And so we could have a neutron break apart into a proton and an electron or beta minus particle.
Okay, yeah.
Because, right? So that's like the reasoning behind it. Now, if they're telling us this is beta minus decay, what we want is whatever we have this decay into, like it would result in there being a beta minus particle, which is going to be mass of 0 and charge of -1. So basically, like what we start off with will decay into the decay product plus the beta uh, product, and the total mass on left and right and total charge left and right should equal one another.
Okay.
So
Let's see. Okay. So beta minus is just, you said it's a loss of an electron.
uh so a beta minus particle is could be thought of as the equivalent of an electron, but if we're having beta minus decay, it's a product,
Oh,
right?
okay.
Mhm. So and so yeah, so we have carbon and phosphorus. So we could start with carbon. So if carbon undergoes, so I could write here, carbon, it has whatever, six like AMU, like or sorry, no, six protons
Yeah.
and a mass of 12, and it'll decay into something plus beta, oops, 0, -1 here. And yeah,
Okay. I'm like,
so we want something with an atomic number that when you add to -1 will give us 6.
oh, um, then I guess it would be nitrogen.
Yep.
I feel like that's kind of I mean, well, I guess like I think it's just confusing because I'm like assuming with the word decay that it would go backwards. But okay,
So the thing that decays is the carbon,
yeah.
right? Because so it's like radiocarbon dating. Like we could do that because over time, and that's like with like half-lives and stuff, right? Like over time, a carbon atom will decay into um nitrogen and a beta particle. And so even so I get what you're saying about if it's called beta decay, it seems like the beta is decaying, and yeah, but it's really something else that's decaying into the beta particle.
Gotcha.
Mhm.
So for the phosphorus, it would be the same thing basically because it's
Yeah, same idea. So phosphorus, which has an atomic number of what?
um 15.
Mhm.
So I guess it'll go to sulfur.
Mhm. Yep. And yeah, you can kind of see like a little pattern here, right? If you have a beta decay, the decay product simply goes to the right by, one like, box or whatever, or column or whatever. You know what I mean, right?
Hmm.
So, and if you had positron emission or positron decay, it would just go left,
I
right?
see.
And then the alpha particle is the only one with a mass. So if you had an alpha decay, oh well, you go left twice because that's the change in atomic number, but you would also have to go down to add the mass. But yeah, but without even like thinking of it that way, you could just kind of think of it being kind of like an equation that you have to make sure like the things on the left equal the things on the right.
Gotcha. Yeah, that makes
Mhm.
sense. Cool. Yeah, I feel like I'll probably just watch more videos on it until I understand.
Yeah, and in the PDFs folder, I have a PDF that's called actually Radioactive Decay, and I explained it in there as well.
Okay. Yeah, I'll take a look at that. Um, and it says compared to normal glycogen, what type of bond, the amount of what type of bond is decreased in Lafora bodies. And that's the branching. So this is the alpha 1,6.
Hmm.
Yes. Okay. Okay, we got through passage 8. It wasn't that bad in hindsight, honestly. I feel like the thing that tripped me up was the figures and the beta decay, but I feel like I'll get it with time.
Yeah, by the way, this is here, right?
Yeah. Mhm. Do we have time to maybe go over this one as well, or is it?
Yep. So I also have an optics PDF that you could look at. Um, but yeah, this one is gonna involve uh well, calculation, of course. So thin lens equation, do you remember that?
Um, no, I feel like I wrote it down the other day. It's like with the DI, or never mind.
Mhm. Yeah, so the O here stands for the object distance, not zero.
Hmm.
It's basically DO, but I just don't write the D part.
Yeah.
So, okay. And they're telling us that the object distance is 3F. So we can plug that in. 1 over 3F plus 1 over I. Then we just solve for, well, I guess we'll solve for the image distance. And then there's the magnification equation, which is image distance over object distance. And so then we'll just, find that.
Yes. So for this equation, I'm solving for I.
So they're asking for the ratio of the height of the image to the height of the object. So we're basically doing I over O, which is basically magnification.
Okay.
Yeah, so you're gonna see that with the MCAT. Like it's rarely ever just gonna be like a plug and chug kind of thing. Like even if it is a plug and chug kind of thing, like they're gonna the actual question's gonna be some variation because they want you to do the calculation, and the second you get the answer to a calculation, they want you to choose that. But it's usually going to be like you get the answer to a calculation and then you do something with it that they ask you about. So like here, like chances, well, I'll let you do it.
Well, I got I feel like well I will equal like 1 over 3f. Or maybe it's 3f over 1 or negative
So
negative.
so what do you so describe to me what you're doing?
Um well I just subtracted the 1 over 3f to the other side so that it will just be 1 over I is
Wait,
equal to
so this is that what you did?
oh yeah, that's what I did. Yeah, my bad.
Okay, now what?
Um I'm like would it be 1 over um negative 2f?
Uh, so tell me like what to do, like what you would do from this step.
Then I guess I would try to condense it. So I would do 1 at 1 over f minus 1 over 3f.
Uh-huh. That's there. Like what would you do? Like how would you do that?
I guess I would have to multiply 1 over f times 3. So then I would multiply the other side um by 3.
So we want them to have so like I mean like if we multiply this by three, like we're gonna have to multiply that by three as well, right? As well as this over here. But I
Yeah.
think what you're trying to say is that we need them to have the same denominator, right?
Yeah. Or
In
maybe 1 over.
order. So the thing is that like so yes, we want there to be a three there, but if we just multiplied by three, we'd have to multiply everything by three. So what we can do is we can multiply by one, which doesn't change anything, but we can write one as 3/3, which then achieves our purpose of giving us the same denominator. But since it's just multiplying, since 3/3 is one, we don't have to multiply every side, by three,
Hmm.
right? Because
Okay.
like if you multiplied by three, right? This would then be 3/f minus 3/3f is equal to 3/I. And we would still have the issue of not having the same denominator, right?
Yeah.
So so what we'll do is we could multiply by 3/3 to get 3/3f minus 1/3f is equal to 1/I, right?
Hmm.
All right, now what?
So then this will be 2 over 3f is equal to 1 over I.
Okay.
Then like do you multiply well now I'm trying to figure out how to get I from the denominator. So I was thinking that you would multiply by 1 to get rid of the 1 over I.
Oh, you could just do like you can like just flip it, or you could do like cross multiply,
Okay.
but we could just flip it. So this could be I is equal to
Three.
3F over 2.
Gotcha. And then we'll put it in the magnification question. And then
Good, good. Because they would have wanted you to go boom, right?
yeah.
So, okay, good. So now, yes, then we can put it into this. So we have 3F over 2 over what?
Says over the object over
Hmm.
3F.
Yep. Now what? So, so I always say that there's no division, there's, but there is multiplication by reciprocal.
Right. So I'm like, okay, so I just multiply it by 3F over 2. So then
So,
just
you would multiply it by the reciprocal. So the reciprocal is 1 over 3F,
yeah.
right? And
Mhm.
these guys cancel. So we have 1/2.
I see.
Hmm. Good, good, good. So, so yeah. So okay, yeah, you need the formulas. That was good. Make sure that this step makes sense.
Yeah, that
And got it. Okay. And yeah, then we solve, so essentially we solve for I, and they want us to go and hit D, but we are being asked the ratio of the height of the image to the height of the object, and then we get 1/2.
can I
Yeah.
take a picture of this?
Yeah, of course, of course.
Cool. Let me just
Mm-hmm.
thank you.
Mm-hmm. So yeah, we're gonna have to wrap up, but so hopefully this was helpful, and um okay. And then I'm meeting, yeah, oh, and you scheduled for Monday.
Yeah, just because I have, well, I don't know, like these next few days, like I'm just gonna be studying because I'm off on Monday too, so
Got it.
might as well just take advantage of the time that I have.
Of course. So you're gonna be taking another test tomorrow, right? Or no?
Second on Sunday.
Sunday? Okay.
Mhm.
Awesome. Good luck on that. And yeah, it'll be um 92
Mm-hmm.
for today. We just went
Gotcha.
like a bit over. So yeah, awesome. Good luck, and I will see you on Monday, okay?
Okay, bye. Have a good
All right.
weekend.
You too. Take care.